Hello everyone, welcome to our second lecture on proteins. So just a recap from last lecture, we looked at our dietary proteins and our recommendations and calculations of intake. We also looked at the basics of amino acid, the structure and properties, as well as the different classifications. We looked on classifications based on polarity, acidity, basicity of the side chains, and our essential and non-essential amino acids. And here's just a reminder of some mnemonics. So remembering all the nonpolar, neutral, and neutral. Grandma always visits London in May for crashing Winston's party. Polar neutral, Sam's teams trust African gorillas. Polar basic, hearts always love, or Hal. And polar acidic, always grateful. Aspartic acid and our glutamic acid, but that's an easier one. So again, for something like the MCAT, pretty much going to have to remember these as well as their structure likely. So you guys, I recommend going back to the last lecture and kind of just going through them, draw them out, and note the differences. So today we're going to be looking at protein synthesis, the protein structures with our primary, secondary, tertiary, and quaternary, also native versus denatured forms, and dynamic versus static proteins. So as we talked about, single amino acids have formed peptide bonds, becoming dipeptides, and then etc. And then these build up to our linear polypeptides, which then form our complex 3D biological proteins through folding and different chemical reactions. So again, amino acids are joined by that condensation reaction where water is removed, joining the two amino acids. 2 or 3 amino acids would be a di or tripeptide, while 4 to 50 would be oligopeptide, and multiple will be a 50 plus would be a linear amino acid, would be a polypeptide. And if we have multiple folded polypeptides from 100 to thousands of amino acids, this is our biological protein in its functional form. So protein structure. So here we have primary, secondary, tertiary, quaternary. So here we can see our linear amino acids. Here we have our alpha helix and beta plated sheet. Here we have our plated sheets and alpha helix folding together, and then this is a bunch of folded proteins together, but we'll get to each of these individually. So as we described, there's our primary, secondary, tertiary, and quaternary. These structures, or protein structures, will ultimately determine how protein interacts with other molecules, like a substrate and enzyme, and therefore they're linked to protein function. So primary structures of protein. So the primary structure is a specific sequence of amino acids in the polypeptide chain. So as we talked about in biology, the ribosome produces this long peptide chain. This is determined by the DNA sequence, then the mRNA sequence, as we discussed. Here we can see uses that coding, and we get that codons. So that's a codon for leucine, and then threonine, and then we build those codons one by one, building that long amino acid chain here. So these primary structures become the basis for all other levels of the structure, and it gives the protein its chemical and physiological properties. These are formed through covalent bonds, which are the strongest bonds in the body. So again, we go from our DNA to RNA to amino acid to our primary protein, secondary, and then tertiary, quaternary. So secondary structure. So the secondary structure is essentially the arrangement of primary structures by hydrogen bonds that form between backbone atoms of amino acids, and not the side chains. So here we can see it can form alpha helices and beta plated sheets with connecting loops. And as I said, two structures are produced, alpha helices and beta plated sheets. So the alpha helix. So the alpha helix is spontaneously formed. The helix is typically approximately 10 amino acids long. There are 3.6 amino acids per turn. So for example, proline ends the alpha helix structure, and this forms a 3D helical cylinder with side chains projecting outwards, and there's no space in the cylinder. So the beta plated sheet. So the beta plated sheet, we can see here our hydrogen bonding. So we have hydrogen bonding between adjacent chains, and these several chains can be involved. It is a three-dimensional structure formed that is formed, and it has crinkles like corrugated cardboard and helps form the initial 3D structure of the protein. You'll see this in a lot of protein structures with the kind of like zigzaggy shape when they use kind of cartoon ways to depict protein. Sorry, I can hear my cat. So our tertiary structure. So the tertiary structure is the most important of the structural levels in determining, for example, the enzymatic activity of protein. So this is when it kind of dictates its function. So a tertiary structure is the folding of secondary structures into compact globular proteins, and these foldings are determined by the interactions between side chains. So this can be either electrochemical attractions or repulsions, as well as different enzymes and stuff like that. So here's an example of some proteins with superoxide dismutase, where we can see these manganese ions. So those are helping form the chain, they form the shape, and we also have here an example of catalase. So one example is that hair straightening disrupts the tertiary structure of the hair protein. So as you may know, hair is made up of proteins, and these disulfide bonds, more disulfide bonds result in curly hair. So through the heat and moisture of straightening your hair, usually with the machine, this makes the hydrogen bonds weaker. And this is essentially the chemical processing of curly hair breaks down protein to straight hair. The hair will return to its original structure. And that's why it doesn't permanently stay straight. So again, there are four main forces that hold the tertiary structures together. There's disulfide bonds, which you can see here. And disulfide bonds which is between two S atoms on adjacent cysteine amino acids. We have hydrogen bonds between partial positive hydrogen and partial negative atoms, such as oxygen. So again, usually you see the H to the O. We also have salt bridges, which are weak bonds between positive and negative side chains. So here it's a weak bond between both positive and negative side chains. You can also have hydrophobic interactions, where it's an interaction between two or more non-polar side chains. So quantitative structure of proteins. So again, we mentioned there's the primary structure, which of the structures represented below is a simple line with just the repeating sequence of amino acids. Our secondary structure, so for example here, a polypeptide backbone exists in different sections of a protein, either as an alpha helix, a beta plated sheet, or a random coil. So again, then our tertiary structure, which is secondary structures folded into compact globular proteins. And we have quantitative structure, which are protein molecules known as subunits assembled into multimeric proteins held together by weak forces. So essentially, a bunch of tertiary proteins form together to form a subunit. So, quantitative structures are essentially the combination of proteins in their tertiary form. Not all proteins have a quantitative structure. So again, many proteins are now functional in this quantitative form and can do their job. They don't need to become a subunit. But when proteins do form a quantitative structures, the tertiary structure is called the subunit. So it's a piece of the other protein, essentially. So example I mentioned before is hemoglobin, which involves several other proteins together. So again, it forms a multi-subunit complex and can be the same or different. And another possibility for functional biological protein. So essentially allows it to have another possible function that it couldn't have alone. So example of this is NADPH oxidase. So here we can see the inactive enzymes where the subunits are dissociated. So these are all our tertiary proteins. But when it becomes active, they bind to this other proteins and now form a subunit, a tertiary protein. So this is tertiary, and we have the active enzyme where the subunits are associated. So just again, learning check. What type of bond is possible in tertiary and primary is covalent peptide bonds. What type of bond is responsible for tertiary or secondary is our hydrogen bonds, which form the backbone. In our tertiary structures, we have salt bridges, disulfide, hydrophobic interactions, and hydrogen bonds. As well, in our tertiary structures, in our quantitative structures, we have the subunits coming together with the tertiary structures, but all proteins are characterized by tertiary levels of structure. So this is what determines its function. It's the most important level. So summary, any protein has the primary, secondary, and tertiary structures. The primary structure is the union of amino acids in the correct order determined by the mRNA and the ribosome. The secondary structure are folds that occur in this polypeptide. So this string of amino acids resulting in the formation of hydrogen bonds in the shape of the alpha helix and the beta plate sheets. Then we have our tertiary structure, which are new folding, which are now new folded, by which secondary structures pass, this time usually mediated by disulfide bonds. Most of the protein is now functional, and some, however, need to join with two or more tertiary structures mounted separately to become functional. When this happens, we have a quantitary structure. In protein, sorry, my cat's going crazy in the background. In protein, we can say that it's formed by two or more subunits. So proteins can be native or denatured. So denaturation disrupts the secondary, tertiary, and quaternary structure of proteins, but the primary structure is unaffected. So at the primary state, this is the stable due to the strong covalent bonds. So we can't break the protein down past primary. We can't break it down into that normal amino acids in normal situations. So denaturation can occur with heating, such as when cooking or when curling your hair. We can denature it from curly straight. So an example, when we cook eggs, we're technically denaturing it. That's why it goes from clear to white. This can also be caused by treatment with acids or bases, and treatment with organic compounds, and treatment with heavy metals. So as I said, a fried egg provides a visual example of a denatured protein. The egg protein is called albumin. So this albumin in its native form is transparent liquid. Cooking it causes albumin to denature, and it becomes opaque and hard. And this is what we eat. So dynamic versus static. So dynamic proteins are not fixed or not anchored to one spot or in the body. So example here, these are just 4 examples, but not all of them. So example, transport proteins, peptide hormones, muscle contractile proteins like actin and myosin, and many enzymes, usually. The static proteins include are fixed or anchored to one spot in the body. So these include various proteins that make up the extracellular matrix. So we have our cell, and they're just on there doing their thing as a receptor or something like that. So let's look, now we're going to dive into some important proteins that you might have heard of. So for example, serum albumin, this is dynamic protein. So serum albumin is a group of proteins that comprise approximately 60% of the proteins found in blood plasma. These are water-soluble proteins, and the function of albumin is the maintenance of osmotic pressure and thus regulation of blood volume. So obviously a very crucial function. There's also it's also involved in the transport of nonpolar substances through the bloodstream, like fatty acids and other lipids, and nonpolar hormones, such as steroid hormones. So another example is globulin, which is dynamic. So globulin is a group of proteins that comprises approximately 40% of the proteins found in blood plasma. Again, it's water soluble, and globulins function in the transport of various molecules within the blood. So here we see globulin in immunoglobulin. So we have more than enough information in this in my immunology series if you want more information about the structure of antibodies. So here we have IgG, immunoglobulin, and then it also is involved in the binding and elimination of specific foreign particles, so from the blood, like antibodies. So again, here's an example I've mentioned before, is hemoglobin. So hemoglobin contains 4 subunits of protein and 1 non-protein heme. So again, this is quantitary. This is found in red blood cells. As we may know, it carries our oxygen. So each of these hemes carries 1 oxygen. So each hemoglobin can carry 4 oxygen. So the 4 subunits are bonded by weak non-covalent bonds, and function is the transport of oxygen. So in comparison, so we know this is a tertiary quantitary structure. In comparison, myoglobin, which has a similar function, is quaternary. So here we can see, essentially, myoglobin is the muscle equivalent of hemoglobin, and only contains 1 subunit of the protein in heme. So sample question. So hemoglobin we know is quantitary, while myoglobin is tertiary. So another example is insulin, which is a dynamic protein. So here we can see insulin in multiple forms, a monomer and hexamer. So we'll talk about these later with GLUT4 and how glucose is uptake through insulin, but insulin is a peptide hormone produced when blood glucose levels are high, and it regulates carbohydrate metabolism. It's very important. And as you probably know, you've heard of insulin in diabetes. So insulin attaches to a receptor on the cell surface, usually GLUT4, And the main function is it triggers glucose uptake into the cell, allowing, so here you can see your insulin receptor. But anyways, so binding of the insulin receptor stimulates GLUT4, and then we have, here we can see our glucose entering the cell. So other examples are dynamic peptide hormones, glutathione hormone, which is a peptide hormone, somatotropin, which is a peptide hormone, and many more. So looking at the muscle specifically, we have our actin, myosin, troponin, tropomyosin, which are the contractile proteins. So actin are thin filaments, which you've probably heard of, and myosin are thick filaments. So here we got our actin, very thin, and we have thick myosin heads here. So troponin, which is found on actin filaments, confers calcium sensitivity to muscle cells, while tropomyosin binds to molecules of actin and troponin and to regulate the interaction of actin and myosin. So again, the function of these is muscle contraction. So we can see here the myosin heads bind to here and they pull, causing that muscle contraction. So another example is collagen, which is an example of static. So again, this is a very structural protein. So collagen has 3 strands of procollagen cross-linked via hydrogen bonds. So collagen is the most abundant protein and it accounts for 25% of the whole body protein content. Again, collagen is super important for structural things and important in tensile strength, but it's important in fascia, cartilage, ligaments, tendons, bones, and teeth, and again, its main purpose is tensile strength. So one example of this is collagen injection. So we see here, before after, we can see almost the smoothing. It's replacing that structural bit. So again, collagen can be replaced in small amounts by injecting into areas such as the mouth. Another example is elastin, which is a very important protein, which is very static. Its main character is elasticity, so this is very important in certain things like our heart, our skin, our lungs, things that need to expand and then return to its original structure. So again, it's very important in our arteries, our lungs, our elastic ligaments and cartilage, our skin, and bladder. So example, your bladder fills with urine. When you have to go, it doesn't stay expanded, so when you urinate, it returns back to its normal structure. That's due to its elasticity and the rugae in the bladder. So example of this, not to throw any shade at Arnold, but this is just an example of how the elastin gives your skin its elastic properties, but the elastin properties and our elastin proteins degrade with age. So we can see that it's lost with aging and skin can sag as a result, unfortunately. So next we have our proteoglycans, which are also another functional protein, which are static. So proteoglycans are found in many tissues, including the blood vessels and skin. So they're essentially formed of carbohydrate chains called glycosaminoglycans. We can see here. And also a central protein core and several sodiums. So proteoglycans are especially important in cartilage and it form carbohydrate chains which trap water through hydrated gel. So this is where the glycosaminoglycans essentially allows it to keep that liquidy kind of thing and absorb liquid. So again, it's mainly involved in hydration. I recommend you look up some stuff about glycosaminoglycans. It's really quite interesting. So here's another example. Again, but it's just an example of how poor lifestyle choices such as smoking and excessive alcohol consumption can hasten the loss of these extracellular matrix proteins such as collagen, elastin, and proteoglycans. So even though you're not as old as some other people, you can actually start to see the loss of these proteins can actually make you age faster than you actually would normally. So again, I guys hope you found that interesting. I'll see you guys in the next one, and that's the end of our protein lectures. Take care.